This is a model of the human ovary. What you see on the right side over here is the suspensory ligament, which contains ovarian arteries and veins. Then you see over here the ovarian ligament, which connects the ovary to the uterus. On the very outside you can see these tiny cells. This is a germinal epithelium. And then inside of that, this white layer is called the tunica albuginea. The outer part is the cortex, the inner part is the medulla. You can see around the cortex there's these tiny little yellow cells that contain a little egg inside. These are called primordial follicles, right? You can see them over here as well. During development and ultimately puberty, where the oocytes start to develop and the ovarian follicles start to develop, it starts to go through changes. You have these primordial follicles over here, and then they turn into primary follicles, which contains a primary oocyte on the inside. And then you got a secondary follicle right here, which you can see additional layer of follicular cells there, and then of course a larger oocyte. Once that matures, that gets to the tertiary follicle or mature follicle. Graphian follicle is another term you would use. This one right here is shown during the ovulation process. This is the ovulated egg, which is a secondary oocyte. Once it's ovulated, then this mature follicle will start to transform. It's going to start to fill in with blood, and the antrum fills in with blood. That's shown here, and it ultimately becomes a corpus luteum, this structure. Corpus luteum is important because you'll see in lecture this is producing estrogen and progesterone. Corpus luteum doesn't stay around forever. It ultimately degenerates into the corpus albicans, which you can see several remnants of that in this model. Okay, so that should be the structures you should focus on in lab.